Okay, welcome to part 2 of Lab 2. In this lecture, I'm going to teach you how to do the calculations for parallel dilutions. You should use these slides in conjunction with Lab 2, part 2, narrated lecture, and along with the Lab 2 Prelab. If you're going through the Prelab and you find that you're having trouble with parallel dilutions, you can consult this lecture. Hopefully it will help. All right, so first let's define what a parallel dilution is. A parallel dilution is a technique for making a single diluted solution of a specific desired concentration. And what you're going to do is you're going to take a solution that's already prepared of a certain concentration. We call this a stock solution. So you're going to take your stock solution and you're going to mix some of that with some water to make a second solution that is more dilute and is of a specific concentration. Now you can figure out ahead of time mathematically what to do in order to make your second concentration or your second solution of a specific volume and a specific concentration. So this is a pretty powerful technique and one that is used very frequently in labs. So let's look at the formula we're going to use for this technique. Good old C1 times V1 equals C2 times V2. Let's talk about it, see how it works. All right, first of all, the C's in this formula stand for concentration. So that's how strong the stock is and how strong the second solution is. And because this is concentration, the units that will be used for the C's are either going to be molar, millimolar, or micromolar. So molarity of some kind. Now your V's stand for volume, how much stock you're going to use and how much the final volume of your second solution is going to be. So since it's volume, the units it's going to use are liters, mils, or microliters. Now when I work with the C1V1, C2V2 formula, I think of the one side as being what I have already. So C1 is going to be the concentration of my stock solution. V1, I don't know. That's the volume of stock solution that I want to use to make my other solution. So I don't know how much to use. That one's going to be an unknown variable. The C2 side is what I want to make. So the C2 is the concentration of the solution I want to make. And the V2 is the volume, the amount of that solution that I want to make. Now in our class, you will always be provided with the concentration of the stock, the concentration you want to make, and the volume that you want to make. However, the V1 will be left blank. That's your unknown variable. But algebraically, if you can fill in 3 of these variables, you can solve for the 4th. So you're going to be solving for V1. And one more note about the units in this technique. Unlike the previous technique, it's fine if you use a variety of different units here, but there are a couple of rules. All of the C's that you fill in need to be in units of concentration, so molarity, millimolar, micromolar. And all of the V's, you need to make sure those are volume, so liters, mils, and microliters. One of the biggest mistakes that students make with this technique is putting a volume number in where the concentration should be and so forth. So make sure you keep your concentrations and your volumes straight. The other thing you need to know about the units in this technique is that the units, you can use any units you want pretty much as long as they are the same on both sides of the equation. So for instance, let's look at this one down here. Clearly it's X'd out, so it's not okay. My C1 is in molarity, molar. My C2, however, is in millimolar. If I leave it like this and solve this equation, my decimal point is going to be in the wrong place, and I'm going to use too much or too little solution. So what I'm going to have to do here is either convert this millimolar to molar or convert the molar to millimolar so that they are in the same unit on both sides of the equation. All right, so let's look at an example of C1V1 and C2V2. Let's say you want to make 200 mils of a 200 millimolar salt solution, and you have a 1 molar stock solution available to use. First thing we want to do is organize our numbers. So my C1 is going to be my stock. My C1 is the 1 molar solution. My V1, I don't know. That's what I'm trying to figure out, how much of that stock to use. My C2 is 200 millimolars. Now again, since that's in millimolars and my C1 is in molar, I need to convert one to the other. In this case, I've chosen to convert this number into molar, and that converts into 0.2 molar. My V2 is going to be 200 mils, and it's just fine that it's in mils. Okay. So now that I know what my numbers are, I want to fill them into this equation, which I've done down here. 1 molar times V1 equals 0.2 molar times 200 mils. Now, let's do the algebra. Let's look at the top of this equation here to start. My 1 molar times V1 equals my 0.2 molar times 200. How do I solve for V1? Well, V1's over here on this side. What I want to do algebraically is get it by itself. So to get it by itself, I need to get rid of this 1 molar over here. How can I do that? Well, I'm going to divide both sides of this equation by that 1 molar. When I divide it here, it cancels out. I can divide it over here. Now, when I divide this side by the 1 molar, the molar units are going to cancel out. And so that means I'm going to be left with mils at the end of this problem. So my answer will be in mils, and that's fine. It's going to tell me how many mils of stock to use. All right, so when I calculate this out, 0.2 times 200, divide that by 1, I get 40. So my V1 is 40 mils of stock solution. I'm going to use 40 mils of stock to make this dilution. Now with this technique, I've got 2 ingredients. I've got a stock solution and I've got water. So this begs the question, how much water should I use? Well, there's 2 ways to figure this out. First way I think is the easiest. You're going to take that stock solution, you're going to pour it into a graduated cylinder. In this case, it's 40 mils of stock solution. And you're going to add water to it until it comes up to your final volume. In this case, you were trying to make 200 mils of solution total. So you just add water until it comes up to the amount you want. You don't want to take your 40 mils and add 200 mils to it, where you're going to add too much water and make it too dilute. So remember to use that graduated cylinder technique to combine your stock with your water. Now, if you wanted to figure out mathematically how much water to use, you can do that too, and it's very simple. What you want to do is subtract the amount of stock solution you're going to use from the final volume you want to make. So there's 2 ingredients, stock and water. You want to make 200 mils of solution. So you just subtract the amount of stock that's in there. And that means whatever's left over has to be the water. In this case, it's 160 mils of water. And you can combine them separately if you want into a container. Mix them together. All right, at this point, I'd like you to pause the video, try to work this problem out, and then we'll go over the answer together. Okay, so go ahead and pause. All right. So in this problem, I'm asking you to make 0.2 liters of 0.5 millimolar salt solution from a 0.5 molar stock. How am I going to do it? Well, I'm going to use good old C1V1 equals C2V2. First, let's organize the numbers. My C1 is going to be my 0.5 molar stock. My V1, I don't know. That's what I'm solving for. Now, C2. C2 is in millimolar. I want to make a 0.5 millimolar solution. So since that's in millimolar and my stock is in molar, I need to convert one to the other. So in this case, I decided to convert it to molar. So I ended up with 0.0005 molar, which is very, very dilute. And that's okay. My V2 is going to be the volume I want to make. So that's my 0.2 liters. All right, I fill these numbers into my equation. I solve for V1. And look at this teeny little number I've ended up with here. My V1 is going to be 0.0002 liters. Now, technically, that's correct. But when you go to actually make this solution, you're going to find it hard to measure that tiny amount with a vessel that measures in liters. So probably what's best to do here is to convert this liters number into a smaller unit, such as mils. So that's the same thing as 0.2 mils. Or you could even convert it into microliters. That's 200 microliters. And we definitely have measuring devices that can measure 200 microliters or 0.2 mils. All right. Now let's consider the second half of making the solution. How much water are you going to add to make the solution? So pause the video, see what you come up with, and then we'll discuss it. All right, so let's see how much water you should add. All right, so in your problem, you were asked to make a 200 ml solution, which is the same as 0.2 liters of solution. Now, since you're probably going to measure your stock in either mils or microliters, I recommend that you convert your final volume to either mils or microliters. In this case, I converted it to mils. So I'm going to take my 200 ml final volume, subtract from it my 0.2 mils of stock that I'm going to add, and that tells me that I'm going to add to my stock 199.8 mils of water. So it's going to be a lot of water and a little bitty tiny drop of solution of stock. All right. Okay. So if we were going to make a solution that is that dilute, there's probably actually another technique we would use called a serial dilution. And I'll teach you that one next. But just as an example, this works. All right. So that's parallel dilutions. Hopefully you understand it a little better now. You can now go to pages 4 and 5 of your Prelab and complete those pages. If you need help with serial dilutions, please consult part 3 of this narration. Thank you very much for your time.